The cell membrane acts as a barrier between the inside of the cell and the outside of the cell. And yet there are things outside of the cell that need to get in and things inside the cell that need to get out. So we need to look at how cellular transport works. The cell membrane is selectively permeable. That means some things get in, but not everything gets in. There are two kinds of transport. Passive transport requires no energy expenditure by the cell. It happens because of normal physical processes. These include simple diffusion, facilitated diffusion, osmosis, and filtration. Active transport does require the cell to spend energy. These are solute pumps and vesicular transport. In vesicular transport, endocytosis is bringing material into the cell. Exocytosis is removing material from the cell. Passive processes require no ATP. They are driven by a concentration gradient. We're going to move downhill. We're going to move from an area of higher concentration of a molecule to an area of lower concentration of that molecule. How fast a molecule moves depends upon its size and its solubility. Smaller molecules move faster than larger molecules. Simple diffusion follows the rules of passive transport, downhill from higher to lower concentration of the molecule. Because we're going through the cell membrane, simple diffusion is based on the size of the molecule, smaller moves faster, and the solubility. These molecules need to be lipid soluble to dissolve through the lipid cell membrane. Simple diffusion is nonspecific. Anything that is small enough and soluble enough will enter the cell, whether the cell really wants it to or not. Simple diffusion is a relatively slow process because it's driven by molecular movement. In simple diffusion, if we were to put this dye pellet into this beaker of water, molecular movement in the dye pellet would cause the molecules of the dye to start to leave the pellet and enter the water. Over the course of time, the molecules would evenly distribute themselves in the beaker of water. We would call this equilibrium. Facilitated diffusion still follows the rules of passive transport. We're still going from higher to lower concentrations of the molecule. But we're going to use a carrier channel or a carrier protein to move the solute. Because these carriers and channels are protein, they're going to be somewhat specific. Proteins are involved in molecular recognition. Also, facilitated diffusion occurs faster than simple diffusion simply because there's a doorway for these molecules to get through. Here you see a protein carrier. It has a precise fit for a molecule, and we are still moving with the concentration gradient. There's more of these molecules outside the cell than inside, and so they're using this carrier to get in. We use this for large molecules that can't dissolve through the cell membrane. Here we have a channel. In this case, the protein has a hole in it. Small molecules can get through this hole. Sometimes the protein is charged, and that allows different charged ions to get through. The specificity here has to do with the size of the hole. Osmosis also follows the rules of passive transport, but this time we're interested in where the water is. We're moving water across the membrane. Osmosis comes into play whenever the solute can't move across the membrane. That is, the membrane is impermeable to the solute involved. Water will move across the membrane to the higher solute concentration to try to equalize the concentrations. Water is constantly moving across the cell membrane, but when there is more water movement in one direction or the other, we will see the size of the cell change. We call this the tone of the cell. And so solutions are described relative to their tonicity, that is, their ability to change the shape of the cell. In an isotonic solution, the concentration of the solute is the same inside and outside the cell. 9/10% sodium chloride is the equivalent to normal intracellular osmolality. We have water constantly moving across the cell membrane, but there's no net movement of water, and so the cell does not change its size, shape, or tone. In a hypotonic solution, the solution is less concentrated than the cytoplasm. 5/10% sodium chloride is less concentrated than 9/10%. Neither sodium nor chloride can move readily across the cell membrane, so diffusion cannot equalize this process. We're going to have to move water. We're going to move water into the more concentrated solution. So water will move into the cell. And because our cells do not have a cell wall, only a relatively fragile cell membrane, if enough water goes into the cell may rupture or lyse. In a hypertonic solution, the solution outside the cell is more concentrated. 0.2% sodium chloride is more concentrated than 0.9%. Again, remember ions do not freely diffuse across the cell membrane, so we're going to have to use water to take care of this inequality. Since outside the cell is more concentrated, water will leave the cell and the cell will shrivel up. If we're talking about a red blood cell, we call this crenation. Literally, the cell is dehydrating. Now, these are red blood cells in different kinds of solution. A red blood cell is normally this size and shape, so in an isotonic solution, there's no change in the size or shape of the cell. In a hypertonic solution, water is going to leave the cell. The cell will get smaller, it will shrink up, and because this is a red blood cell, we call this crenation. In a hypotonic solution, the cell will take on water. This means the cell will swell up and potentially could rupture. Filtration is driven by hydrostatic pressure. Here there's more pressure on one side of the membrane than the other, and both the solvent and the solute are forced through the membrane. The only limiting factor as to what goes through the membrane is the size of the pores in the membrane. Molecules have to be small enough to fit through the pores of the membrane. Filtration is used in the body when we make urine. One form of active transport are the solute pumps. Here we're moving molecules against the concentration gradient. We're going uphill. We're going from a lower concentration of the molecule to a higher concentration of the molecule. This is going to require energy. So the cell will use ATP, and to make sure we get the right molecule, we use carrier proteins so that we recognize the molecule and bring it across. ATP will change the shape of the carrier protein so that we can push the molecule in one direction or another. The sodium-potassium pump is an example of active transport. Our carrier protein is set up to pick up 3 sodium ions. As those 3 sodium ions nestle in place in the protein carrier, ATP is split, and this provides the energy to change the shape of the protein carrier. This will dump those sodiums out and also open up 2 seats for potassium. Potassium will come into the carrier protein, and because this change of the protein shape was very temporary, it will go back to its normal position, which is open to the inside of the cell, and it will pump those potassiums in. The protein carrier is now available, and if there's ATP available, we can pump 3 more sodiums out and 2 more potassiums in. Vesicular or bulk transport is also driven by ATP. Endocytosis is taking something into the cell. The cell membrane will move around the particle and engulf it and form a vesicle known as a phagosome. This will bring the particle into the cell. Exocytosis is the reverse of this process. It's pushing the material out of the cell. So here we have a cell taking in something by endocytosis, and we have our phagosome. And once this is in the cell, several things can happen to it. It can simply move across the cell and be dumped out by exocytosis. It can be merged with a lysosome and the material digested for energy, or the membrane may return back to the plasma membrane to refresh the plasma membrane. There are 2 types of endocytosis. Phagocytosis is when solids are brought into the cell. This is the equivalent of the cell eating something. Pinocytosis is when the cell takes in liquid. This is the equivalent to the cell drinking.